In Lab 9, students are going to learn about cell division, specifically the two processes of mitosis and meiosis. As with every lab, there is a set of learning objectives that we want students to master by the time they complete the lab. Students should be able to describe the different events within the cell cycle. We'll be going through those. We want to talk about differences in regards to cell division with mitosis between animal cells and plant cells. Students will be identifying different stages of mitosis using plant cells, specifically the onion root tip, and animal cells using whitefish blastula. We will also be using models, both plant and animal models, to look at the different stages of meiosis. We're going to learn how to calculate time spent in each stage of mitosis, and students will be expected to be able to do that calculation on both lab quizzes and lab exams. We will describe the different events in meiosis. We're going to show you some images of meiosis. We're going to do some modeling of the different stages of meiosis, either by using beads or by drawing those different events out. And then finally, we want students to be able to compare these two different types of cell division. So we want to be able to compare mitosis and meiosis. In this lab, we have a fairly robust list of keywords that students need to familiarize yourself with. So please make sure that you're taking the time to do that. So in this lab, again, the primary focus is to understand cell division. And cell division can be defined as the process where one cell is going to split into two new cells. This process is observed when we injure ourselves. So for example, if you get a paper cut, you expect cell division to happen to repair that injury. We also know that cell division happens when an individual grows. So as we get larger, cells divide, which helps in that process. One of the things that we also need to understand is that when cells divide, there's a series of steps that have to happen. So we're going to have to talk about how we have to make additional genetic material. We have to duplicate our DNA. We have to replicate our chromosomes. We're also going to have to make sure that we increase the amount of cytoplasm and cell organelles that we have, so that when the two daughter cells are produced, that they are functional cells. Now, we're going to start first by talking about mitosis, and then we will move into meiosis. So, organisms like you and I are going to do both mitosis and meiosis. Mitosis is really a process that's going to be done with what are called our somatic cells. Excuse me. Our somatic cells are those cells that are not going to become gametes. And when we use the word gametes, we're talking about your eggs in your sperm. Meiosis is going to be the process that organisms that reproduce sexually are going to use in order to generate their gametes, in order to generate their eggs and sperm. And so the cell division processes of mitosis and meiosis are different, and they're not done by every organism. Again, certain organisms that divide asexually. For example, yeast can divide by the process of mitosis. One cell becomes two. There isn't sort of this necessity for sort of male and female gametes to come together and fuse to form a zygote. So I do think it's important to recognize that although we're going to talk about these processes, mitosis and meiosis, that not every organism is going to do both. Again, that process of meiosis is going to be a process that's done by organisms that reproduce sexually. So, as I said, we're going to start off first by talking about mitosis, and mitosis is done by your somatic cells. So, again, these are going to be most of the cells that make up your body. Mitosis is going to happen in regards to those cells that aren't going to become your eggs and your sperm. It is important to recognize that Certain cells in the body are amitotic. What that means is that sort of when you reach maturity, those cells no longer divide. So, for example, neurons that make up your brain, most neurons are what we consider amitotic. So that means is if they're damaged, they can't be replaced by new neurons. You will learn more about this. Those students that continue on in anatomy and physiology will learn more about this. Now, when we talk about the process of cell division, we really like to sort of organize it in what we refer to as the cell cycle. And the cell cycle is sort of an ordered sequence that really extends from the time a cell is first made until it itself divides. So when we talk about the cell cycle, we typically divide this into sort of two major stages, interphase and the mitotic phase. Interphase is going to have sort of three subphases. There's going to be G1, also known as gap1, synthesis, and gap2. Then we talk about the second major phase, that's going to be the mitotic phase. This is where we're going to see the genetic material, the DNA, divided equally between the two daughter cells in a process called mitosis. And that's going to be quickly followed by the process of cytokinesis, which is going to allow for the cytoplasm to divide. So we're going to go through these different stages. But before we do that, I first want to talk a little bit about your genetic material. So what's going to happen is in interphase, as I mentioned, we're going to subdivide this into gap1 synthesis and gap2. So in gap1, the cell is going to, and you can kind of see that denoted here on our little diagram here. In gap1, the cell is sort of growing, it's increasing in size, and it's really performing sort of normal cell function, whatever that normal cell metabolic function is. If it's a skin cell, it's acting as protection. If it's a muscle cell, it's important in regards to being able to contract and allow for movement. So again, when we say normal function, it can mean different things depending on the cell that you're looking at. Now, once the cell reaches a certain size, or potentially if there are cells that are damaged, what's going to happen is there's going to be chemical cues in the cells going to enter into synthesis. In synthesis, what's going to happen is our chromosomes are going to duplicate. Now, for you and I, we have 46 chromosomes in our somatic cells. So 46 chromosomes, or oftentimes what we refer to as 23 pairs of chromosomes. Now, those 23 pairs of chromosomes, okay, those 46 chromosomes, they have to be duplicated. So we want to duplicate each of the chromosomes before we're going to enter into the final phase of interphase gap2, where by that time, when we get to gap2, replication will have been completed. Here, cell organelles are duplicated, and the cell is going to continue sort of normal metabolic function. Now, before we move in and we talk about how the DNA, the genetic material, is actually divided, I want to spend just a few minutes talking about what happens during this S phase when the chromosomes are duplicated. So our chromosomes, eukaryotic chromosomes, are going to be made up of something called chromatin. And chromatin is really DNA molecules that are a DNA molecule that's wrapped around little histone proteins. So right here we can see DNA. Again, we expect DNA to be double-stranded, right? And you can see that. What's going to happen is, in order to sort of keep it in a semi-condensed state so that it fits into the nucleus, what we do is the DNA is condensed by wrapping it around these histone proteins and forming this chromatin. So you can kind of see that, right? So here's our DNA. The DNA is wound around these little histone proteins, and it sort of condenses the DNA. Now, the chromosomes can be at different stages of being condensed. It can be very, very condensed. When it is, we can clearly see the chromosomes, or it can be sort of semi-condensed. And when that happens, we can't really clearly see the individual chromosomes. It actually appears to almost be like a ball of yarn, okay? Now, what happens is when chromosomes duplicate, so this would be a chromosome right here. When it duplicates, it makes an exact copy of itself. So here we're going from a chromosome this would be one single chromosome. When we make a copy of this, there's going to be a identical copy of that first chromosome. Now, even though we've made a copy, this represents a copy of this first chromosome, these two what we call sister chromatids are actually going to be held together. Okay, so each chromosome, after it goes through replication and it's made of these two sister chromatids, they're actually going to be held together at a region called the centromere position, or the centromere spot, or the centromere region. So although we actually have two chromosomes, we actually refer to this as one because it's physically being held together by little proteins here at the centromere region. Okay, and I'm going to talk more about that in the next video when we talk about meiosis. But again, what I want you to understand is when you see a chromosome like this is a chromosome that's in the duplicated state. All right. Normally, if the cell's not dividing, you would have just a single chromosome that looks like this. So again, assuming if we just take a quick peek here, we go back, we're in gap 1, right? This cell is doing sort of normal growth, increasing cytoplasm. We've now duplicated our chromosomes. We went from a chromosome in the unduplicated state to the chromosomes in the duplicated state. So now we have 2 copies of each chromosome. We've done gap 2, where we've made more organelles, we've made more cytoplasm, and we're now ready to enter into mitosis and cytokinesis. So this little diagram here, this little picture, shows you each of the stages that make up, each subphase that makes up mitosis. So there's prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. So what's going to happen is during prophase, the chromosomes are going to begin to condense. Now, what do I mean by that? Well, they're going to wind tighter and tighter and tighter around these little proteins, and we're going to actually begin to see those chromosomes. So the chromosomes become much more apparent in prophase. Other things that are going to happen here is that spindle fibers, which are proteins that are part of the cytoskeleton, are going to grow during prophase. And they're going to form a structure called the mitotic spindle. The function of the mitotic spindle is really to stretch the cell and to physically move the chromosomes around during mitosis. Now, during this stage of prophase, we also have these little tiny structures that you're kind of seeing here in blue. These are centrosomes. Centrosomes are going to be present in both interphase and mitosis. Centrosomes in prophase are going to begin, these little centrosomes here, they're going to begin to migrate to opposite poles of the cell. And that's important because these centrosomes are going to act as little organizing centers that help to position the mitotic spindle. So those proteins that are going to be very important in stretching the cell and moving the chromosomes. Now, what's going to happen after prophase is you're going to have prometaphase. And during prometaphase, the nucleus is going to completely break down and it completely disappears. The spindle fibers are going to attach to the chromosome. So you can see our little spindle fibers here are attached to our chromosomes near sort of a middle region of the chromosome, that little centromere region. And they're going to begin to move the chromosomes into the middle of the cell, into the center of the cell. Now, once those events happen, we're now sort of entering into metaphase. Metaphase is going to, in metaphase, what we're going to see is that the mitotic spindle is going to be fully formed. And what the mitotic spindle is going to do is it's going to align our chromosomes on the metaphase plate. Now, the metaphase plate isn't sort of a physical entity. It's really a region that's located in the center of the cell. So keep in mind that, it's not a structure. It's sort of the equator of the cell, if you want to think of it that way. So at this point, each sister chromatid, remember that our chromosomes in the duplicated state are each made up of two sister chromatids, are going to be attached to the mitotic spindle. That's important because when we get to anaphase here, that's what's going to allow for those chromosomes to eventually be, those sister chromatids to eventually be pulled apart. So during that next phase of anaphase, you can see here that the sister chromatids are going to be pulled apart and towards the opposite poles of the cell. The mitotic spindle does this as it shortens, and the cell is also going to, if you can notice the cell shape it's changing here, it's also elongating, it's changing as well. Now it's at this point that our sister chromatids are now considered full-fledged chromosomes. So if we think about numbers, okay, so let's say that in our little cell here, and again, chromosome number is going to differ from organism to organism, but let's say in our little cell here, we started with 4 chromosomes. We would start with 4 chromosomes in the unduplicated state. Then they would go through synthesis and they would be 4 chromosomes in the duplicated state. So we can see that, right? We have our 4 chromosomes in the duplicated state, each consisting of 2 sister chromatids. We still have our 4 chromosomes in the duplicated state as they align on the metaphase plate. And here, when we get to anaphase, you can now see once the chromosomes in the duplicated state, once those sister chromatids have been pulled apart, now we actually double our chromosome number. So if you count them up, 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. That's important because at the end of mitosis, the cells that we produce should be genetically identical to one another, and they're genetically identical to the parent cell that they have come from. So in anaphase, you can see the sister chromatids are pulled apart, right? We've doubled our chromosome number. By the time telophase occurs, okay, so you can see that here in the next image, the chromosomes have been separated and basically moved to opposite poles of the cell. The chromosomes are now going to begin to sort of decondense. They're unwinding into looser chromatin. And you can see that the nuclear envelope is going to begin to appear. It's going to begin to reappear around each set of chromosomes. So again, we've got 4 chromosomes in this, what will be this nucleus, and there will be 4 chromosomes in what will be this nucleus. Now, other things that are going to happen in telophase, the mitotic spindle is going to begin to break down, and at the end of telophase, the cell is going to have elongated, and there are going to be 2 nuclei present. Cytokinesis, which is this final stage here, is a process that really, if we sort of think about it, it actually began slowly back here during anaphase. It doesn't complete until cytokinesis, the final step in cell division. During cytokinesis, the cell is going to divide fully into 2 separate daughter cells. With mitosis and cytokinesis complete, the 2 daughter cells are going to now re-enter into interphase, and the whole process can begin again. Now, one thing to note is that animal cells and plant cells are going to do the process of cytokinesis slightly differently. In animal cells, cytokinesis is going to occur, the splitting of these 2 cells, by using little contractile proteins, almost like a little belt. And those little contractile proteins are going to tighten until eventually you're going to pinch apart the cells and separate them. In plant cells, it doesn't quite happen the same way. Because plants have that cell wall, they have to do this process of cytokinesis slightly different than what we do in animal cells. So to accomplish this, what's going to happen is there's going to be these little membrane-bound vesicles that begin, and you can kind of see those little membrane-bound vesicles. Yes, some sources call them Golgi vesicles, but they begin to accumulate near the metaphase plate. And as the vesicles begin to fuse together, they are going to form what is called a cell plate that's going to run from one side of the parent cell to the other. The vesicles and the materials they carry are really going to form the new cell wall that exists between the daughter cells. So again, we need to familiarize ourselves with these terms because they are bolded, and we do expect that students are able to compare and contrast this process of cytokinesis, this process of mitosis in animal and plant cells. Now, that was a brief explanation of the process of mitosis. In the next video, I'm going to go through what students are going to need to do for their different lab procedures for this lab.